Good morning, Kylie. Just wanted to test the audio out from the room. Can you hear us?
Hey, Steven, this is Chris. I can hear you fine on the webinar.
All right, thanks, Chris.
Hey, good morning, everybody. This is Mike Luisi. I was just checking in with council staff to make sure that my audio is working okay. Let me try that again. This is Mike Luisi. I'm just checking in with staff to see if my audio is working okay.
Mike, this is Carlos Lozano from AKRF. I'm not part of the staff, but you're definitely coming in loud and clear.
Okay, thank you very much.
Well,
I'll go on mute for a few minutes. You guys don't want to hear me coughing and sneezing for the next half an hour.
Good morning. This is Adam Nowalski.
I can hear you, Adam.
Good morning, Mike. Hope you're doing well.
I hope is your son okay? I hope he's doing okay.
Yeah, no, it was pretty much over and done with in 24 hours for him.
Oh, wow.
So just trying to be proactive and respectful of everyone else.
Of course, that's why I'm at my house right now. It's been like 10 days for me and my wife. It won't go away. So anyway, I'm glad he's doing better. Nice to hear your voice.
All right. Yep, thanks. I'll be on mute.
Okay, me too. Hi, for those of you in the room, this is Mike Luisi. I'd like to give everybody a 1 or 2 minute notice before we get the meeting started. So if everyone can start to take their seats, we'll get started in about 1 or 2 minutes. Thanks. Hi, good morning, everyone. I can't see you, obviously. I am not there in the room with you all, but this is Mike Luisi, Chair of the Mid-Atlantic Council, and I'd like to call to order the June 2022 meeting. We have a full plate in front of us today, and I wanted to take a moment before we get started. There's a couple announcements that we want to make. First of all, I wanted to let everyone know, whether you're participating online or you are there in the meeting room, I very much apologize for not being able to be there, but my entire family has come down with COVID over the last week, and I just didn't think it was appropriate, and I'm sure that you can all agree. So I've had, excuse me, I've had a number of discussions both with Chris Moore and Wes Townsend, who is our council's vice chair, and we're going to split some duties up a little bit over the course of the day and into tomorrow. And so I plan to kick the meeting off. And then after our morning break, I've asked Wes to chair the few items that we have on the agenda before lunch. And then when we meet jointly again, well, not again, but when we meet jointly with the policy board of the Atlantic States Commission this afternoon, Wes and I have decided I would chair that. And so we're going to bounce things around a little bit so that you don't have to listen to me cough and sneeze and sniffle for the next 3 days. So I appreciate all the support that I've received from folks that I've spoken with. I tried to call almost everyone, but I wasn't able to get to everybody before the meeting. So thanks for your support, and we have a lot on our plate to deal with today, as well as the rest of the week. So I want to get things moving into our first item on the agenda. But before I do, I want to turn the mic over to Chris Moore, who has a couple announcements to make. Go ahead, Chris.
Chair, good morning, everyone. I'd like to introduce our two newest staff members. I think everyone knows that we've hired two folks, hired them both in May. Sitting in the back is Tori Kettner. Getting feedback, Stephen. Fix that. Anyway, you hear this twice then. So Tori is a spatial ecologist. You'll hear from her shortly regarding the NERA project that she's been involved with. She's been involved with that project for a number of years on a contract with us. Before that, she worked for the National Marine Fisheries Service. She's working out of her home in Brooklyn, New York, and welcome, Tori. The other person in the room that's new is Hannah Hart. Hannah worked for the Florida Fish and Wildlife Commission for I think almost 8 years. So Hannah started on the 16th of May, and she'll be involved with, of all things, SCUP and some other projects as well. So welcome, Hannah. So if you get a chance to introduce yourself and say hi, they'll be here all week. Thanks.
Yeah, thanks, Chris.
And before,
Yeah.
if I can interrupt you, Mike, before
Yeah,
we
please go for it. Go ahead.
Yeah, fix this reverb here before we get anything.
Okay, just let me know when it sounds better.
Okay.
How's that sound now? No, sounds a lot better now.
I don't really hear much from the webinar side as far as reverb, but
Yeah, it's on this side, Mike, but I think sounds good. It's fixed.
okay.
So again, Tori and Hannah, if you didn't hear me.
Yeah, no, we got you. I appreciate that, Chris. And I had the opportunity to meet Hannah. Not that I've known her for years, but at the CCC meeting a couple weeks ago. So, welcome, Hannah and Tori. I'll look forward to meeting you the next time we all get together, and hopefully I'll be in much better shape than I am today. So, but welcome, and I look forward to working with both of you guys into the future. So let me think if there's any other, are there any other? So the only other announcement that I can think of at this time, and I think all of you are well versed in this, is that because I can't be there in person, we're going to operate the meeting almost like a webinar. So hopefully everybody has some form of device that they can use to raise their hands. So I have the participant list up in front of me, and after the presentations, when we do, when we do questions and comments, I'm going to look to that participant list for hand raising. We will offer opportunity and time for the public, depending on time on the agenda, as we get through the day. And so that's how I'm planning to operate. And if, for those of you in the room, Chris, Wes, and others, if something were to, I usually get pretty decent, oh, I just got told we lost my audio.
Test, test.
Test, test.
Here, so.
All right, see if we're back online. Mike, can you hear me now? Mike Luisi?
Yeah, I hear you, Wes. Can you guys hear me from my end?
Yeah, you sound good. It
Okay.
didn't take long, about 5 minutes for me to screw the whole system up. So let's try this again.
Yeah. Okay, so I don't know how much you heard. I talked for a while. I was given some ground rules for the day. I don't know where I left off, but I don't want to repeat myself. What's the last thing you heard from me?
Pretty much hello, and that you're sick. I'm sorry that you couldn't be here.
Okay. Um, yeah, so the other thing that I wanted to make sure that members of the council and later on we'll talk about the same thing with the policy board. We're going to operate the meeting basically in a virtual setting, although many of you are together in one room. If you can raise your hand online, that way I can see the hands go up. And instead of calling on people, it's what we've been doing for the last few months, year, or whatever it's been. So that would be the preferred way to get us through today and the rest of the week. So with that said, I think we've covered our first topic on the agenda, which is technical difficulties, and that's okay. It happens. And thanks, Stephen, for being able to make those corrections. I think the best thing to do right now is to just start diving into our agenda. We have an update. Our first item on the agenda is an update on the Northeast Regional Habitat Assessment products. And we have Jessica Coakley with council staff who is planning to give us that information. So with that said, Jessica, I'm going to turn it over to you.
Thank you, Mr. Chair. I'm actually just going to introduce the topic. We have a number of our team members that are going to be presenting today. Tori Kentner of council staff will present, as well as Chris Hack from Monmouth University, and he's working at Monmouth cooperatively through an agreement with NOAA Sandy Hook lab as well. Another team member, Michelle Bachman from the New England Council, is online, as well as Laurel Smith from NOAA Fisheries. So our team of 5 has developed this presentation on the Northeast Regional Marine Fish Habitat Assessment. We've provided some background behind tab 1 of your briefing materials. So today really is just, because we've only got a half hour, it's a short preview of some of these habitat science products that we're going to be rolling out later this summer. You'll be receiving a more detailed presentation this fall when the council can consider whether they'd like to take up an amendment to integrate all of these habitat science products into your essential fish habitat designations, consider them as you develop habitat areas of particular concern, and other needs related to an essential fish habitat review or redo that hasn't occurred for a number of years, but are required to occur every 5 years. Just so you're aware as well, another product that will be coming to you will be an SSC summary report. Last week, a subpanel of Mid-Atlantic and New England SSC members reviewed some of these products that are in development. I think it was really well received. So we're really looking forward to seeing that report and sharing that with you. So with that, I'm going to hand this over to Tori and Chris to present on NERA. Thank you.
Hi, everyone.
Yeah, so
This is Tori
before
Kettner.
before you guys get started, I'll look to you, Jess, maybe to, since I'm not there, to help, if you want to stop in between the different presentations for questions, or do you want to go through all the presentations before we take questions and comments? What's your preferred option there?
Questions at the end, please.
Questions at the end. Perfect. All right, sounds good. Take it away.
Okay, sure. This is Tori Kettner, and I'm going to give you a quick overview of the NERA project along with Chris Hake. So our goal is to describe and characterize estuarine, coastal, and offshore fish habitat distribution, abundance, and quality in the Northeast. We have 4 main action items. So we're looking inshore at fish habitat assessment, so this distributions and abundance, as well as some status and trends. Also habitat vulnerability, including response to climate change, spatial descriptions of species habitat use offshore, and creating habitat data visualization tools. So our scope is in the Northeast US, so from Maine to North Carolina, and also from inshore and offshore, so mean high water all the way to the shelf slope. We have 65 focus species, which you see listed here. So a lot of familiar faces there. Summary of products. So we are wrapping up this project right now, or at least getting to our first kind of rollout of a lot of products. So we have the data inventory. So we went through all of the data sets that we could find in the Northeast and tried to pull in fish data, habitat data, anything that we could find that would really help illuminate what's happening in this area. We looked at habitat use. So we created species profiles as well as some stage-based single species and joint species distribution models. And then we also created an inshore habitat report. Climate vulnerability, we have a species habitat matrix and climate vulnerability narratives. For habitat data visualizations, we created a Shiny app, which is an interactive place that users can explore the data in these areas at state and regional scales. And then we're also working with a number of partners for sharing these products. We'll have a number of papers and publications that come out of this on the modeling products, as well as an R package, and there's other things in development as well. So here's just an overview of a lot of these products. You can see a lot of great work is coming out of this, and Chris and I will dive more into these throughout the rest of this talk. Okay, I'll hand it over to Chris now to run through the joint models that he's been working on.
So first, I'm going to start off by giving just a kind of quick review of, of our rationale that sort of informed how we're going about the modeling work. You guys all know about fish habitat and what it is, but on the organismal level, it's a function of several things. The organism's innate physiological tolerances, what sort of salinities and thermal environments they can function in. It's also a function of their ecological requirements, things like shelter from refuge from predators, access to resources, and those things change over multiple life stages. As animals grow, their requirements change as well. So it's a complex order, and on top of all that, they're responding to dynamic environmental factors that change on monthly, daily, yearly timescales. So trying to make sense of habitat is a complicated thing. When you move up to the community level, it gets kind of more complicated because you're not only now thinking about an individual animal and how it responds to the environment, but you're also looking at how animals respond to each other, how they push and pull each other around in space. And on top of that, dispersal limitations. How can an animal reach a location to inhabit it? So all of those different factors can interact to produce positive and negative correlations in the abundance of different species. And that is something that's not often dealt with on the modeling side of things. So just a few quick examples about how animals can influence each other's use of space, for example, and habitat. So there's fundamentals like competition, species that have similar environmental requirements that might compete can exclude each other from space. Sometimes predators track the distributions of their prey. Non-consumptive effects as well. Prey might alter their use of space as a result of predator abundance. Some people call that fear effects. And then you can have other more complex things like social interactions between competitors that might share a predator and could co-occur together to share information about that predator as a means of sort of reducing their potential risks of predation. And all these things can scale up and to be visible at broader scales. They don't just happen at the individual level. So all of these things kind of informed how we went about working on the habitat models. We took a stage-based approach, breaking species down into different life stages to try and get a better picture of stage-specific niches or ontogenetic shifts in habitat use. We are modeling with both a joint species model that kind of tries to take into account some of those biotic interactions that I just talked about. And we're trying to make a point of using dynamic, temporally varying covariates, predictor variables, whenever we can to reflect the dynamic nature of the environment that these fish are living in. And we're also trying to identify predictor variables that have a real direct relevance to the ecological function of animals. And I'll talk a bit more about that in a bit. So you guys all know, again, how do we survey fish? We can't just drain the ocean and see where they are. We sample very sparsely. In 1 year, 700 surveys, 700 tows for the NIMS bottom trawl survey, sample less than 1%, less than 0.1% of the seabed. So how do we take that sparse data and make inferences about habitat across a larger area? That's where species distribution models come into play. Species distribution models try to estimate the habitat niche of an organism by relating its observed abundance in, say, the survey data to a range of environmental variables, the sort of environmental filters that we touched on in the other slide. So you take your observed abundance and look at how a species responds to temperature or to salinity or to other variables, and use that to make predictions about where you should find it in space or how many animals you would expect to be in a given location. And so you can take that and those niches that you estimate and use that to kind of draw inferences over a broader space. But also when you model, you end up with residuals. Your model is never 100% correct. You overpredict in some places and you underpredict in other places. And when you're working in a single species context, those residuals just get chalked up to error. And some of them are indeed just chance stochastic error, but some of those residuals can also be informative. So when you're working in a multi-species context, you can look at the residual patterns across species, essentially, where do you overpredict and where do you underpredict after you've accounted for environmental predictors, and how does that correlate across species? So if you consistently find that some species occur together more often than you would expect, given their environmental preferences, that might be hinting at an underlying ecological interaction. So joint species distribution models are a way of trying to model that residual covariance. And in doing so, you produce, theoretically, more realistic predictions about community assemblage. So one of the downsides to joint models is that they're relatively new and they're very complex and computationally dynamic. And I'm going to spare you the details, but I'm fortunate to have some collaborators that are better statisticians than I am, who help to figure out a way of doing this faster and better in a way that accommodates really big data sets like the NIMS trawl surveys that we're working with now. So moving on from there, we're applying that modeling methodology right now to the bottom trawl survey data. Right now, we've got almost 100 species/stages of animals included in the model. These include the nearest species of interest, different functional groups, demersals and pelagics, as well as just common species that occur frequently in the survey, and other taxa that are known to be prey or maybe important components of the community on the shelf. So the model is trained on 15 years of data, and then we're testing its predictive performance on 5 years of data. We are combining the spring and fall surveys and using right now 13 different predictor variables that include temperature at different temporal scales, salinity, both of those on surface and bottom. Other things like sea surface height and some other correlates of depth that I'm also going to talk about soon, along with some complex things that I'm not going to bother you with. So predictor variables, correlates of depth. So water depth is a commonly used and really informative predictor. It's been around for a long time, and it's measured by the survey, and it's great. However, in most cases, it seems like it's really a proxy for a whole bunch of other things that range from temperature to the underwater light environment to the strength of water movement at the seabed. And so there's evidence already of species modifying their use of depth as a means of tracking, say, cooler water temperatures as the oceans warm. So using depth as a predictor over the long term might not be that informative if that's not really the proximal thing that's driving their distribution. So we made a point of trying to find other predictor variables that could be related more directly to animal function. So among these, like as I just reviewed, sort of bottom stress, it's a measure of the intensity of water movement at the seabed. So it's inversely related to depth. Wave energy dissipates the deeper you get. And although you can have things like bottom currents that also cause that. So we're including that as a predictor. We're also including two metrics that measure sort of the underwater optical environment. One, PAR, photosynthetically active radiation, is really just a metric of light intensity. How bright is it? So shallower waters tend to be brighter, deeper waters tend to be dimmer, but shallow turbid waters get dimmer faster than shallow clear waters. And then you have this other metric called hue angle, which measures the spectral distribution of light. So it's not necessarily how intense the light is, it's what frequencies of light, what wavelengths are arriving at a certain location. And so that tends to capture the gradient between coastal waters, where you have a lot of suspended particulates and things look redder or yellower, out to more oceanic waters. And so by including all these things, we're hoping to find ways of accounting for the effects of depth, but that have greater direct impacts on the animals. So how does it work? The model seems to be performing really well. It does a better job than most of the other alternatives. It has a higher predictive skill and the comparable levels of error. And so that's a good thing. Happy about that so far. So just reviewing some of the things that we get out of this model. So we can look at species relationships with the predictor variables. What are the factors that are driving species distributions, and how are they responding to each of these different factors? Kind of creating a metric of the species niche for each of the different predictors. I'm including, and here we've got looking at the optical niche for summer and winter flounder, which is kind of an interesting comparison. So summer flounder undergo this seasonal migration from deeper to shallower waters, and their optical niche on the left reflects that. You can see that yellow is like a higher probability or abundance is a stronger effect, positive impact on the likelihood of an animal being there. The blue represents a lower one. So you can see that the summer flounders span both that on the x-axis there is that coastal to oceanic gradient, and they're using that whole region, whereas winter flounders tend to stick to more coastal waters in terms of the underwater light environment. We can also look at the importance of different predictors and how they, for each species. You can see for summer and winter flounder here, they both have similar patterns, but for example, summer flounder tend to respond more to surface temperatures, where salinity has a greater effect on winter flounder. And you can also see that juveniles and adults of the two species tend to respond similarly to the same suite of predictors. Another thing that comes out of this that you wouldn't get out of single species models are these residual correlations. So this is the relationships between species that are left over after you've accounted for the effects of the environment, after you've accounted for their environmental preferences. And so here you can see, for example, really strong correlations between adults and juveniles of each species. That makes sense. You'd expect that things, either dispersal limitations are going to lead to adults and juveniles occupying similar places, as well as maybe social interactions. And then you have weaker correlations between those two species, but they're also positive. And a couple other sort of interesting little things. And so these things, this residual correlation matrix gets used when you're generating community level predictions. So that rather than just predicting based on the environment, you now take those residual correlations into account, and you can say if two species don't co-occur frequently, that will come out in your predictions. You just get these more realistic predictions of community assemblage, basically. And then quick example of some predictions. Flounder, summer flounder, you can see pretty clear seasonal migrations. Not so much for winter flounder, which is pretty characteristic. And you can see pretty clear evidence of stage-specific differences in habitat use as well. So next steps here are to include some additional predictor variables. Right now, we don't have benthic habitat characteristics in the model, things like sediment characteristics and structural complexity that can be important, particularly for demersal species. So we're going to bring those into the model. We're going to finalize the results, work on visualizing them and making them available via portals and the Shiny app that Tori's going to share with you. We're also going to try and take these models and use them to drive long-term predictions of how habitat use might change in the future as water temperatures change. And finally, we're also hoping that we can include some response data from other surveys, for example, NEMAP, to try and fill in gaps in coverage that exist in the nearshore region. And with that, I'm going to pass it back to Tori.
Okay, so I'll share my screen. It says that I'm not a host. Okay, got it. Okay, so I'll give a real quick overview of the Shiny app that we've built. And so this is online. You can see the link up top. It's nira.shinyapp.io/data explorer. And just that alone will get you here. So feel free to check it out. We are officially launching sometime in July, so this is kind of still a work in progress, but feel free to go to the link, play around with it. What you see here is our homepage, so you can click on any of the different modules that we've built. There's a regional view, a bay view, a species view, a model view, and some of the metadata and reports that we've created. So regional view, you start here, select your surveys that you want to look at and your years, hit go. You'll get an overview of top 20 species by abundance and biomass. There's also a species list, so you can search for a specific species by typing it into the box, or a certain survey in the box, and then you can filter that way. You can look at top abundances and biomass for all of the trawls and seine surveys. There's an analysis of the seasonal temperature and salinity, both surface and bottom. Same thing for the bays. So this is a similar, all the same data sets, but you're just filtering either by a certain area. So Long Island Sound, for example, we'll start with. And you can turn on and off surveys that are in that area, again, filter by years. And you get your top 20 abundance and biomass for those particular surveys in that particular bay. And then again, you can look at the species list. One thing that's different here is you get a cluster analysis. So this is basically showing you species that are caught together more often. So if they're closer together on this cluster, then they've been caught in tows together more often than species that are farther apart. Again, seasonal temperature and salinity graphs there to get an idea of kind of the conditions across time. Then we'll go to the species view. So here you can look at an individual species. So I'll look at scup, for example. The first plot is a relative abundance. So what we were looking at is kind of trying to update the Elmner figures. And so you get the abundance across different months. You can again change the year ranges. And then this is broken down into different salinity zones. So tidal, fresh, mixing seawater, as well as adult and juvenile. Occasionally, we don't have that data for all species, so you might see some no stages or some NAs. That's because we don't have salinity zones or stages for those. There's a distribution map there, so that's showing you the abundance of each species. And then we also have some species reports that we've created. So there's species profiles, which are background and life history. This is the kind of classic essential fish habitat designation reports. And then we've also created some climate vulnerability reports. So I think right now we have that for Atlantic cod. Again, here you can search for species or you can type in a species. So there's our species narrative. And so this is some climate vulnerability information. For models, so this is the work that Chris has been working on. So we've got our distributions for summer flounder and winter flounder. Currently, we'll be adding to this as we continue on. Some covariate importance, species correlation. So all of that will all be hosted here. So the idea kind of this app is just a way for our team to share all of the work and all the products that we've been working on. Something in particular, actually, that I should mention that's special about this app, in addition to all of our work, it also is the one place where you can see a lot of these inshore surveys in one spot. So different state surveys, they kind of have their own portals where they share their data, but this is one spot where you can see all of these surveys in one place at one time. So I think that's really useful, especially in the inshore region. I haven't really seen that before. So what we created, so everyone can understand where this data is coming from, we created this metadata tab, and so you can go through any of these different surveys or habitat data, anything, you can take a look at the data. So we created these one-page overviews, so who collected the data, a little bit about the resolution, overview, methods, caveats you should know about, how to access the data, those type of things. So this is kind of like your one-stop place to understand the data that we're using in this. Reports, you can check back here as we get our reports rolled out. This is the trawl survey comparison. So if you're looking at maybe Maine and Massachusetts trawls together, you want to know how similar the methods are, you can look at this. You can see maybe one's using a 3.5-inch mesh, while another one has a 2-inch mesh. Just gives you a little more background on how the different data sets are collected. And then to look at similarities in time, both on months and years, I created these two figures. So the top figure shows you all of the different surveys and the years that they have been collecting data. And then the bottom shows you the months where they collect data. So not all surveys collect data at the same time of year. Then last page is a little bit about us. So you can kind of see all of the amazing people who have helped with this work. And so that's your real quick overview of the Shiny app. This is, like I said, the work in progress. It's online now, so feel free to review, send me comments, questions. And then we should officially launch this sometime in July. Okay, is that everything, Chris?
So, a few slides.
Oh, is it a few more slides? Okay. Okay. So I'm just waiting for it to be shared. Okay. So there's a few applications for these near products, so how we're going to use them. So one is for essential fish habitat. So we're hoping that the information that we're using can kind of make better EFH text descriptions and maps in the future. There's also the state of the ecosystem reports. So the habitat and climate change information can help on managed species. And then also some of the single species assessments. We've already started working them into even some stock assessments. So I did a little bit of work with the butterfish stock assessment, and we're hoping we can do more of that in the future as well. So helping with stock health and giving some background information on some of those environmental drivers. So there's several data portals that are already publicly available. So we're going to work some of our products into that, as well as hosting it on the Shiny app. So you can see some of those other portals that are there now. I gave you guys the overview of the Shiny app. So there's the link there at the top in case you need it still. And then lastly, this is our timeline. So we started in summer of kind of 2019. We're working now through the very end of the project. So this is finalizing and distributing products. So you can see a lot of work went into this over the last few years. You know, we started off just looking at some of our goals, prepping all the data, running the models, and then finally we're in the last stages of preparing everything. So again, just thank you to all of our teammates. You know, it's not a single person or even a few people. We've got a lot of help from a lot of people. Okay, we'll take questions now.
Yeah, thanks, Tori. Thanks, Chris, for your presentation. No fault of your own, but we're a little behind schedule. But I'm going to take a couple questions or comment at the same time. I have Dewey Hemmelwright with his hand up. Dewey, go ahead.
Yeah, thank you. Thank you for your presentation. My question would be, given the steering committee action teams, the wealth of knowledge of all the folks here that's representing and distribution of maps of fisheries and different things, is there anywhere possible that these teams all put together could do something of an overlay of VMS data to show where fisheries are happening based on that VMS? Because I'm seeing in some stages where BOEM has brought forth the call area for Central Atlantic and solicitation and comments, particularly to pelagic longline, that the only vital information that will actually show potential for this gear is to start where the gear started at, sit. HMS has only brought forth where the sit is. It doesn't show where the gear ends up being retrieved at once it's fished, and that would be through VMS data. And also, you look at other fisheries that have VMS, and it would be very helpful because as we're sparsely boxing off areas of the ocean, it would be good to see VMS data, which is accurate to the vessel and the different gears and fisheries to actually show where that's taking place. So until that's actually showed on a map, all this other stuff is interesting and great, very helpful. But that would be the really genie in the bottle to show where this is actually happening right at the vessel level. So if that in the future could be done and incorporated into what y'all have here, I believe it would represent a more accurate picture and which would be vital to the fishermen and industries of all types to show what's happening. Thank you. I don't know if that was a question. Well, maybe a question and a comment.
No, thank you. That was a really good suggestion. Yeah, we've really only been working with state and federal surveys at this moment. We haven't worked with any vessel data. So that could be something that we definitely should consider building into like Shiny 2.0 or Nero 2.0. I think that's very interesting.
And just to follow up, because not everybody's required to have a VMS, so it's not like you got 10 million people. I'm sure it's a few thousands or something like that probably. So it might be interesting on how to unlock the key to get to that data. And remember, we don't care about the confidentiality to show any of that. Just track lines and maybe having track lines of different gears that's operated throughout the area. Thank you.
Yeah, thanks. I appreciate that, Dewey. That was kind of a, going back in the past, like a Rob O'Reilly kind of question, but it's all good. I'm glad you were able to make your comment. I'm going to go to Mike Simpkins next. And well, let me do this. Is there anyone else from the council? And Mike, you're sitting in for the Science Center, right, today? Yeah, go ahead, Mike. And then Kate, I'll come back to you in just a second, but go ahead, Mike.
We're getting him a mic.
So that was a lot of effort to get me a mic for me just to say, I am sitting in for the Science Center today, and this is a really impressive body of work. Y'all should be really proud. This is a great compilation and really useful. Thank you.
Yeah, thanks, Mike. I'm used to seeing John's name up there, but no, thanks for being there to represent the Science Center today. Let's go to next to Kate Wilkie. Go ahead, Kate.
I got it. Okay, thanks, Mike. I've had the opportunity to see this presentation several times in different settings, so I don't have any questions at the moment, but I just wanted to acknowledge, as Mike just said, what a great body of work, very impressive, and I think it's cutting edge science, and happy to have supported it along the way, and thanks for your work on this.
Thank you.
Yeah, it's a lot of really impressive work, and my notes indicate that we'll be getting a more detailed presentation at our October meeting. And so we'll look forward to that as well. Is there anyone else from the council? I do have one more hand up on the webinar from Carlos, but I'd like to see if anyone else from the council wants to ask any questions or provide any comment before we move on. I've got about 2 minutes before I want to kick off the next agenda item. Okay, I'm going to go to Carlos Lozano.
Yeah, this is Carlos Lozano from AKRF Inc. Just want to say that I agree with everyone else. This is great work, very important. I have a couple questions, hopefully not too long. Hopefully I'm understanding correctly that you use CPU-E data from the trial survey as abundance for the dependent variable and the GAN models. I was just wondering, have you considered using the binary presence absence dependent variable in the models? Thinking about some of these lower abundant species that probably aren't sampled as regularly in the trial than others. My second question was, have you considered using the PCA analysis to generate orthogonal relationships among species using the model residuals? Thinking about, have you, maybe this will help find like functional groups or similar species that might have relate to each other in terms of habitat?
Hey, this is Chris. Yeah, thanks. Those are both really good questions and points. So to your first point, yes, we are actually, I didn't get into the details, but we're actually using a hurdle model because the data is so dispersed for a lot of species that we can't fit a straightforward negative binomial or something. So there is a presence absence component, and then we model abundance conditional upon presence. So we do have there's a binary response as well as the count response. For your second question, yes, we've also looked at doing ordinations of the ONSE the same way you would with like a latent variable model, doing ordinations, spatiotemporal ordinations as well with the with species basis function coefficients, the way they're sort of loadings against the basis functions, and those things they're kind of in the pipeline.
Great, thank you.
Okay, thank you, Carlos, for those questions, and thanks, Chris, for the answer. In the interest of time, I think it's time that we move on. Excuse me, to our next agenda item. And so, Tori, Chris, thank you very much for your presentations. We look forward to follow up at our October meeting. And Jessica, thanks for introducing the topic this morning. We're going to move ahead to the next item on our agenda, which is an aquaculture update. We have Jose from council staff here to provide that update, and there is a motion on behalf of the committee that we'll need to consider as the council. Carlos, if you could also put your hand down, that would be helpful. Thank you very much. So I'm going to turn it over to Dr. Jose Montanez to provide us this presentation. Go ahead, Jose.
Thank you. Thank you very much. So today we're going to be reviewing two draft documents that are under tab number 2. The first one is the aquaculture background document, and the second one is the aquaculture policy document. We're also going to briefly review the recommendations that came out of the Ecosystem and Ocean Planning Committee meeting on May 10. And then, as Mike indicated, we're going to move into the aquaculture policy document approval. So first, I'm going to give you a little bit of background of how these two documents were developed. Council staff used the existing aquaculture background and aquaculture policy document that were developed by the New England Fishery Management Council. The New England Council used a multi-layer process to develop those two documents, and they had input from staff from New England, GARFO, the Mid-Atlantic Council, and the Northeast Fisheries Science Center. In addition, when the policy document was developed, they also had a lot of meetings and input that came from the Habitat Committee, PDT, and the Habitat AP. And the council adopted the aquaculture policy document in 2020, December of 2020. So we have used similar approaches like this in the past. For example, the New England Fishery Management Council, they took the habitat policy document that was developed by our council, and they made some minor modifications to reflect the needs of the New England Council, and they passed that, they implemented that policy document in an expedite fashion by using first our documents. So we have done this before in multiple occasions. So now I'm going to move into the aquaculture background document. And this, again, this is in tab number 2. So this document is intended to provide an overview of aquaculture activities in the Mid-Atlantic region and information related to the potential effects on Mid-Atlantic Fishery Management Council species and habitat. So we go into defining what aquaculture is, and you see like about 3 or 4 textbook definitions of what that means. And then we talk about the different areas where aquaculture occur, inshore waters, offshore waters, on land. And then we also review the species that are currently being cultured in the Mid-Atlantic region. So if you go to table 1 of that document, you see a listing of the species, the type of culture method that is used, and the relative economic importance of those species to each one of the states where they're being produced. The information that you see on table 1 came directly from state agencies. The document also provides an overview of the current aquaculture permitting and authorization process in the region, and a review of the process in place that is designed to consider and avoid or minimize potential negative effects on Mid-Atlantic Council managed species and habitats. And the document also briefly summarizes how aquaculture operations might interact with other human activities, including fishing. A lot of the information, permitting information that came at the state level came from state offices, aquaculture and fisheries offices. So the document also has a pretty good description of what are the positive and negative impacts that could occur to species in our region and habitats as a consequence of aquaculture. For example, positive impacts you have heard before, and you know, aquaculture has been shown to be positive in carbon and nutrient sequestration, improve water quality, coastal protection, habitat provisioning, and things like that. And this is mostly associated with shellfish and algae culture. But researchers have also identified several potential impacts, negative impacts to fisheries and EFH from marine aquaculture, like finfish and shellfish operations, with water degradation as being one of the major concerns from aquaculture operations. The document includes reference to various best management practices, and this is important as they describe how best management practices combined with advances in production methodology have limited the risk of environmental impacts for marine fish aquaculture. So we have included in the document a lot of pretty good information regarding best management practices. Now, moving into the aquaculture policy document, basically the scope of activities that we're trying to cover in this document, the principles that we have listed, they go across various phases of an aquaculture operation, such as permitting, construction, and the operation of the aquaculture facility itself. The document also mentions the overarching Mid-Atlantic Council habitat policy and the Council's role in EFH and HAPC designation and consultation. The document states that the Council is seeking development compatible with the protection of Council-managed species and their habitat, and with commercial and recreational fishing activities. The document also emphasizes that an early and after approach is something that we should be looking at, and that we also need to consider the importance of cumulative effects of multiple aquaculture operations and things like that. And then the document specifies the audience for this policy. And the primary audience for this document is the Council itself as we engage in these consultations. Secondary audiences are NOAA fisheries, federal agencies, state agencies, fishermen, aquaculture developments, and the public in general. So now I'm going to move into talking about, briefly discuss some of the aquaculture principles that are included in the policy. So the first principle deals with site and design projects in the context of ecosystems, functions, and services. And here's something that is important, is that we have to consider the distribution of a specific sensitive habitat, look at EFH, HAPC, and consider all these areas when developing aquaculture projects. To adopt operational practices that reduce environmental effects. And here we're talking things about secure gear maintenance, the use of native and naturalized species when possible, are some of the things that are mentioned under that. The third principle is consider cumulative effects. Again, multiple aquaculture operations, you don't just look at them in isolation, you have to look at them combined to assess what the impacts of those operations could be. That contribute to the local coastal economies, and that they're developed in the context of other sectors, policies, and goals. And here we have to consider the distribution of fishing grounds and the potential for adverse interactions. And one of the recommendations is to start with pilot scale projects and then see how that works before moving to larger scale projects, and to use high quality data to understand other uses and how those other aquaculture operations can interfere with fishing, commercial fishing, recreational fishing, or other uses of the ocean. The sixth principle talks about clear and ongoing communication, how important this is. And we're talking about here communication between the council, the commercial and recreational fishermen, developers, regulating and consulting agencies and members of the public. But it's good to get involved early in the process as you can tackle things if there are problems. And the last one is the collection of baseline scientific data. And we're talking about how this should be something that is required or necessary as part of the permitting process. And we're talking here about things like comprehensive seafloor survey, robust hydrological water quality surveys, and other environmental surveys as needed. And then under this specific principle, there is also several research plans, but one of them that is very important is that support ongoing monitoring, and so we can assess how the aquaculture operations are impacting, or if they're impacting the habitat and the species that are found in those areas. The reason I have this last principle here in italics is because this was added to the policy document by the committee in May 10. And some of these things that are in this principle, you're already finding other sections of the document, but I think that it's a good thing to include it here in the document because it captures all that baseline scientific kind of needs in one place. Furthermore, this is also part of the permitting process. So when you're doing permitting process, this has to be also done. But I think that it's good that the policy spells it out. So where do we go from here? The EOP met on May 10. They suggested some minor edits to the documents for clarity. They added Principle 7, as I already indicated, and they also passed a motion where they recommended that the council approve the draft aquaculture policy as modified by the EOP committee. And this motion was approved by unanimous consent. So for the next steps is if the council approved this policy, we're going to include the policy with the council policy and process documents that we have that you see on our website, where we list all the different policies for different items that the council deals with. And obviously this policy could be revised in the future if needed via a council action. In addition, we're also going to put the background document in the aquaculture web page that we have at the council, so people can have access to that information as well. We're also going to continue to have a dialogue with NOAA Fisheries and on how to best work together to provide input on specific projects and on aquaculture planning in general. And then we're also going to continue to work with our partners at the New England Fishery Management Council on issues of shared interest related to aquaculture. That concludes my presentation, Mr. Chairman.
Thank you so much, Jose. I really appreciate it. And thanks for setting up a slide on approval and next steps. As I mentioned before, there is a motion for council support of the ecosystem and ocean planning motion. But before we get to the motion, let me see if anyone from the council has any, does anyone have any questions at this point for Jose?
No.
I see, let me look who that, Mike Pentney. Go ahead, Mike.
Thanks, Mr. Chair. And sort of half question, half comment, Jose, on the principle number 7, which was the new addition. I think, in principle of the principle, I agree and acknowledge the need to collect data and have clear sense of what information will be collected prior to permit approval. But I just worry that the principle 7, the way it's written right now, is overly prescriptive in terms of the specific, very specific data types and surveys that would be required for every project. And as you noted, there's information that's required as part of the permitting process, but we're not in control of those permitting processes of other agencies. And should their requirements change, we could end up with conflicts in terms of what's called the specific types of data and surveys that are called for in Principle 7 versus what the permitting agencies are actually requiring, which might be project specific. And so I just worry that we're setting up, setting the stage for having an overly prescriptive Principle 7 that could be in conflict with the requirements and provisions of the permitting processes that we're trying to, in some ways, support. So that's sort of a question, if you can interpret that as a question, like, was there some consideration of writing Principle 7 to be more general and supportive of the surveys and data requirements that the permitting agencies would impose without being so prescriptive?
Yeah, the way I see all these principles, these are suggestions that the council is making to the individuals that are planning on to develop an aquaculture operation in our region. So I think that just guidelines. It's not like I understand what you're saying, and I think that you make perfect sense. There are different agencies that are going to have different requirements for a lot of the permitting that they control. I have seen a few aquaculture pre-application, for example, pre-application requests for 2 aquaculture operations, and basically both of them kind of like indicated that they were going to do this type of baseline analysis. So I understand what you're saying. I don't see a conflict, but if the council wants to revise that language, I think that is something that can be done.
Mike, go ahead, Mike.
Yeah, thanks, and I appreciate that. Just as a follow-up, I should have mentioned this. Other question is whether this is intended to apply to all aquaculture projects within the region, state and federal waters, or just in federal waters. And the reason I mention that is I think, we could all assume that or expect that the sort of data requirements and survey requirements for a large-scale project in federal waters might be different from a small seaweed, a farm in an abatement, far inside of state water. So just curious about that.
It was federal waters. Yeah. So in principle, if somebody wants to get a permit to have an aquaculture operation in federal waters, they're going to be dealing with federal agencies. Now, the states, they can get involved under the Coastal Zone Management Act, and also, there is an overlay in there. Now, all other aquaculture operations that are going to be developing state waters, then you have the states are involved in that, but you also have federal agencies that are involved in that also are part of the permitting process. But for this, my intention was just federal waters. Yeah.
Are you okay with that, Mike? Do you, anything else before I go to Eric?
I'm all set, thanks, Mr. Chairman.
Okay, thank you. All right, I'm going to go to Chairman Reed from the New England Council. Go ahead, Eric.
Yeah, thank you, Mr. Chairman. And I understand Mr. Penny's concerns. You know, nobody likes conflict, especially between partners. But you know, these documents, they're not set in stone. They're living documents. They're made to be guidance, basic guidance. And if one shoe doesn't fit all, these documents can easily be reviewed and changed. So I'm not overly concerned about where we're headed today. So thank you.
Okay, Eric, thank you. Thank you for your comment. Is there anybody else? Excuse me. Is there anybody else from the council that would like to ask any questions or before we get into the motion that we need to discuss? All right, seeing none at this time, if I could ask staff to bring up, or maybe Jose or somebody from staff to bring up the motion that was made on behalf of the Ecosystem and Ocean Planning Committee. And I'm going to turn to Kate to make that motion once it's up on the screen.
Stephen, we don't see it on the webinar.
Yeah, it's not popping up. It's not on my screen here.
I might be able to read it that far away, but okay, maybe it's coming up on the webinar now.
Let's give it a second, Kate. We'll get it. We'll get it up. I want everybody to see it, you know. There we go.
Okay, do you see it now, Mike?
I can see it now. Yep.
Okay, so on
I
behalf
can't, it's not, it's small, but I can see it. Yep. Go ahead.
on behalf of the Ecosystem and Ocean Planning Committee, move that the council approve the draft aquaculture policy as modified by the EOPC on May 10th, 2022, and reviewed by the council today.
Okay. Thanks, Kate. As chair of the Ecosystems and Ocean Planning Committee, Kate Wilkie is making that motion on behalf of the committee. It does not require a second. Kate, did you want to speak to it at all?
Well, I'll just say that, you know, during the committee, we had a good discussion and review of the policy, and we were working from the extensive work that was done with the New England Council to build off of their policy. And so, you know, I think this is a strong policy, and there was support for the small changes that we did discuss making. So
Okay.
that's all.
Yeah. Yeah, I totally agree. And I'm sorry I wasn't able to make that meeting, but yeah, I agree with your comments. Is there any discussion on the motion by members of the council at this time? Any discussion? Is there anyone from the public that would like to address the motion at this time? If you could raise your hand on the webinar. Okay, seeing no hands coming up, is there any objection to the motion? Any objection? Okay, seeing no objection, the motion carries by unanimous consent. Thank you, Kate, for your work on that with the committee. And let me go back to Jose. Is there anything else at this point, Jose, that needs to come before the council regarding this aquaculture update?
Well, I just want to mention that now the policy document now that has been approved, we're going to post that on our website with no modifications. However, I'm going to make a few modifications to the background document. I got some comments on Friday from North Carolina regarding some language that will enhance the permitting and process from that state and some other things. So there are going to be minor modifications, but it will not impact any of what we have done today. Thank you.
Okay. Excellent. Thanks for that. Okay, so we're going to wrap up that item on the agenda. I know that I'm trying to make up time. You guys are doing a great job getting us there, but I would like to let's just take a quick break and come back at 10:30 for our next. We have 3 more items before we break for lunch. Our next presenter is presenting remotely, so staff indicated that we need to get them set up for that, and we'll come back at 10:30 and we'll pick up the New Jersey Ocean Acidification Monitoring Network. Thank you very much.
Hi, Grace. This is Mary. If you want, you can do a quick audio test.
Hi, Mary. Can you hear me?
Yep, sounds good. Thank you.
Great, thanks.
Two-minute warning, 2 minutes. We'll come back to the meeting. All right, everyone, break's about over. Let's everybody find their seats, please.
Okay, welcome back, everyone. I'd like to dive into our next item on the agenda. And as I mentioned earlier, I know we were having some technical difficulties with communication. I did mention that after this presentation, I'm going to turn the chair over to Wes Townsend, who's our council's vice chair, to see us through lunch, see us to lunch, and then I'll pick up after lunch. So just to kind of mix it up a little bit, and Wes agreed to that, and we thought it was a good opportunity. So I'm going to go ahead and introduce the next presenter. We're going to get a presentation on New Jersey Ocean Acidification Monitoring Network. The presenter is Dr. Grace Saba from Rutgers University. And Dr. Saba, are you there and ready to start your presentation?
Yeah, I'm here. Can you hear me and see
Excellent.
the screen?
Yeah, I can see the screen. Like you, I'm virtual today, so please go ahead and get started anytime you're ready.
Great. Thank you so much. And thank you everyone for having me and giving me the opportunity to speak with you today. We've been, I'm at Rutgers University. I'm a professor there, and I've been working with the New Jersey State for a few years now, 2 or 3 years, on their development of their ocean acidification action plan. And this is just kind of one of the most recent efforts we've been working toward. And I just want to say that I know this is very specific to New Jersey, but there are several other states in the Mid-Atlantic and in New England that are working on similar initiatives. They may not be acidification monitoring networks, but they're working on, multiple and different efforts to advance ocean acidification research, monitoring, and an action plan for policy. So this is just a little bit of a flavor from New Jersey anyway. So kind of a primer on ocean acidification, and the reason why we're doing all of this is that ocean acidification, we have this increased carbon dioxide in the atmosphere through human activities, and the ocean absorbs about 1/3 of that carbon dioxide. And so what's happening is as the carbon dioxide is being absorbed by the ocean, there's a complex set of chemical reactions that act to reduce pH, which is the measure of acidity in the ocean. And since the Industrial Revolution, atmospheric CO2 has increased by about 40%. And if you look on the right, that atmospheric CO2 is the red line here, if you can see my cursor. So it's increasing atmospheric CO2, and that's been measured at the Mauna Loa in Hawaii since 1958. And then we have seawater samples that have been collected now since the late 80s, early 90s. And with increasing atmospheric CO2, you can see this green line of increasing seawater carbon dioxide, and then the simultaneous decrease in pH here with the blue line. And so we're seeing that drop in pH. We've seen about a drop of 0.1 pH unit, which doesn't sound like a whole lot, but pH is on a logarithmic scale. So it's a pretty rapid decrease in pH in seawater since the Industrial Revolution, and it's equivalent to about a 28% increase in ocean acidity. Oops. There we go. And we expect this to continue with business as usual practices. So on the left, we have atmospheric carbon dioxide under different emission scenarios, the red being RCP 8.5, that's kind of business as usual. And so we're projecting a doubling of carbon dioxide in the atmosphere by 2100, and that would be equivalent to a drop in pH, an additional drop in pH of about 0.2 to 0.3 pH units by the year 2100. Looking at that on a map, you can see drops in pH all over the oceans, global ocean. And that would be equivalent to about 100 to 150% increase in ocean acidity. And again, it's not just about pH. The ocean has a natural buffering process, which acts to remove carbonate ions because they want to bind with the hydrogen ions to form bicarbonate. But what happens is the carbonate ions are really important in organism development, specifically with shell structures and things like that. So that form calcium carbonate structures. And then that lowers the aragonite or calcium carbonate saturation state. So it makes it harder for organisms to build those skeletons. So we have that kind of traditional ocean acidification occurring, which is the ocean absorption of that carbon dioxide. But there are also a lot of local factors that can act to exacerbate or actually eliminate acidification. I'm going to just give you some of the details on how it can act to exaggerate ocean acidification in local systems, near shore coastal systems. So we have fresh water coming in, and that fresh water is lower in salinity. It's typically higher in dissolved inorganic carbon and lower in total alkalinity, which acts to kind of increase the effect of ocean acidification. It's less buffered and it can't handle changes in pH very well. So that fresh water can act to enhance ocean acidification. We also get runoff with that freshwater input that has a lot of nutrients, which can promote phytoplankton blooms in the coastal ocean. That in the surface water can actually act to increase pH. But then once those cells start to die and sink out, all the respiration that occurs in bottom water can act to decrease pH and cause acidification in the bottom waters. And then we also have upwelling that can occur on the coastal shelves where you get deep water that's lower in pH upwelling towards the nearshore system. So it's high in variability and complexity in these coastal shelf systems that can act to exacerbate ocean acidification. Just some observations to show you some real data here. These are seasonal transects off the coast of New Jersey, and what we were really trying to see was, are there seasons that we need to be concerned about? And if you look, these are cross-shelf transects from a glider that can measure from surface to bottom, has a pH sensor on it. So these are pH data. And what we found in the data, and what we're consistently finding in the data, is that we're seeing low pH in bottom waters in the summer. It's a very important habitat for a lot of species. Just here I have shellfish species here, but there's a lot of other finfish species and things of concern. And we also see a lot of freshwater near shore, and that's pretty much in every season, maybe except the winter time. So there are things that we're now observing and things that we can think about in terms of maybe where we might need to monitor more. This is a data product that we put together looking at just summer bottom pH, and now we're realizing that summer might be the most affected season in terms of acidification. This was in the most recent State of the Ecosystem report. This one was in the Mid-Atlantic. We also have this figure in the Northeast State of the Ecosystem report. But this is bottom pH data from 2007 to present. So it's plotting all the available quality controlled data that we could find. And just kind of things to highlight is, we see this lower bottom pH in Long Island Sound, off the coast of coastal New Jersey, and then in the bottom waters of Jordan and Wilkinson basins in the Gulf of Maine. So those are kind of areas that we're thinking we could monitor more. And why do we care? It all comes down to how the organisms respond to this. This was a meta-analysis done by Christy Croker, where she looked at mean effective size, which is essentially, it's the logarithmic response ratio from a organism that was in an acidified treatment versus a control. And so a negative response indicates that they're responding more negatively in the acidified treatment. And she combined a bunch of studies looking at different organisms across a whole spectrum of types, and then looking at different processes, so things like survival, calcification, development, abundance, and found that there's a lot of negative responses in these organisms. Now, these were all done on lab-based studies, and that's really kind of what we've had to go on in terms of determining responses of organisms. And I'll talk a little bit about that in just a few slides. But we're also seeing now impacts of reproduction, behavior, even swimming ability. And so we're starting to think of how those might interact to affect things like biotic interactions, so things like predator-prey interactions and biodiversity and entire ecosystem responses. And then there's been a lot of research effort going into, can these organisms acclimate or adapt to acidification? So a lot of research going into this now. Specific to the Mid-Atlantic, we did a literature review of ocean acidification focused studies on important organisms in the Mid-Atlantic region. The study was looking at, I think we found 18 species relevant to the Mid-Atlantic in about 59 studies. comprised of different types of species, and all of these species are managed by one or more of the fisheries councils. We looked at a wide range of response variables, and what we found was that about 65% of these organisms had a negative response to acidification, and some had no effect. And again, these are based solely on lab experiments. So through, thinking in terms of the drivers of acidification in different regions, as well as the dependence on the living marine resources that we have in the region that might be susceptible to ocean acidification. There was a study done by Ekstrom in 2015 that kind of tried to see where there would be social vulnerability because of climate change, and this is specifically looking at ocean acidification. And the areas that highlight in this kind of maroon color are the highest social vulnerability. And that was, South Jersey is one of those, also getting here in the Massachusetts area and into the Northeast. And again, that's due to a combination of the drivers that cause acidification and the dependence on those organisms. And so in terms of New Jersey specifically, we're predicting that New Jersey could be at high risk of economic harm from ocean acidification conditions because of these factors. And specifically, southern New Jersey counties rank second in the U.S. in economic dependence on shellfish, which could suffer from increasing ocean acidity. And so this was a concern to the state. And so acidification was included in that Executive Order 89 signed into law by Governor Murphy. And so that kind of spurred off all of the recent policy efforts that I've been working on with the state of New Jersey, focused on ocean acidification and developing an ocean acidification action plan for the state. And so what we've been working on is the state formed an ocean acidification team. The team engages with Rutgers, particularly myself and Jean Herb at the Blaustein School of Policy. We're kind of, some of the earlier efforts were learning ocean acidification action planning from other states, kind of just figuring out what other states are doing and how that might be adapted to New Jersey. And then working on elements for the ocean action plan. And kind of in terms of the process of us figuring all of this stuff out, one of the key takeaways was that if we could develop a comprehensive statewide monitoring network, that would really help in terms of figuring out how the state can make decisions. And so that's kind of where we're at now and what we're doing. And so there were three tasks that we've been working on over the past year. And that was really to, we convened a workshop in November, so that was part of task 2, but trying to kind of outline an approach for developing an acidification monitoring network. And right now we're in task 3, and that's why I'm presenting to you today. But we really wanted to engage stakeholders, figure out how we can partner with stakeholders in a proposed monitoring network. And so we want to obtain feedback from different types of groups on, anything from where do they think we should be collecting samples in terms of where they're fishing and things like that. But also, are there ways we can partner and collect chemistry data along with the activities that are going on? And so we've started kind of writing a draft on how we would develop a statewide ocean acidification monitoring network. And kind of the first thing that the state should do is convening an ocean acidification task force or working group, or just some group that can kind of get through these pretty massive tasks. And that's to inventory the current monitoring assets, assessing gaps in the monitoring, and then prioritizing and filling those gaps to improve the network. And again, this is really in order to make decisions for the state. So those are three really big tasks. Luckily, we've had some progress with the first two just from earlier efforts through the Mid-Atlantic Coastal Acidification Network, or MACAN, which I'll talk about in a second. The other tasks for developing a statewide ocean acidification monitoring network are very, basically very logistics based. So I'm not going to cover those today, but I just wanted to include them so you could see other things that we were thinking of, because there's a lot of logistical things we have to get through in order to make this happen. A lot of that has to do with ensuring data quality control and how we handle the data and make sure that we can actually collect the data. So kind of going back to inventorying current monitoring assets, the Mid-Atlantic Coastal Acidification Network had an effort where we looked at where carbonate chemistry samples were being collected in the whole Mid-Atlantic. And this was done, I think we started that effort in 2018. And so this is kind of an example of a map, and these maps are available on the MARCO Ocean Data Portal. And they have metadata associated with them, you know, what types of data were collected, how often, those types of things. And when you first look at the map, it looks like our region is pretty well covered, but most of this green is from cruises that only come in every 4 years or so. You know, and then we have some data that's being collected that's only being collected in over time and not over space. So there were some needs that we kind of thought of initially that need to be met through some future observing. And that's a higher sampling frequency, monitoring across the salinity gradient, so from the rivers to the open ocean, measurements of multiple carbonate chemistry parameters, and that's really because you can't measure things like carbonate saturation state directly. Those have to be calculated by having several variables measured. And so if we could measure a few more variables, we could calculate that. Higher resolution depth profiling measurements. A lot of the measurements we have so far are only at the surface. And with our new understanding that there's acidification events happening in the bottom waters in our region, we need to have more measurements at depth. Observing ocean acidification with other stressors, so things like hypoxia, low dissolved oxygen, warmer temperatures, all of these things can happen together and are happening together. And so we need to measure them at the same time. Co-locating biological response monitoring, so that's a big one. That's so we can measure carbonate chemistry, but if we don't know how the organisms are responding to it, how useful is that? And again, this kind of goes back to the laboratory measurements where, pretty much the only response data we have for organisms is from lab-based studies that are very controlled. And we need to see more studies that are looking at how organisms are responding in their natural environments under changing conditions. And so those are the types of things that we need an observation system to look at. We thought of some opportunities to fill gaps. This is definitely up for more discussion, and an OA task team would hopefully help us fill in some of that. And some ideas here are just using buoys that are already out there to add sensors to. We have New Jersey has a lot of glider deployments, these underwater robots that we can add sensors to, so it's kind of enhancing those systems. But partnering with industry groups is one that we're really interested in doing, because you can get the data that's relevant to the stakeholders, but you also can co-locate some of that some of those chemical measurements with biological measurements. And so that's where I'm at, and I'm hoping we have a little bit of time for questions and just for some discussion, because we're really just trying to reach out to different stakeholders now to see what the interest and willingness is to maybe modify existing efforts to optimize monitoring in New Jersey, but also other states as well are very interested in this. And then obtaining just feedback on just logistical considerations for maybe adding some measurements to existing operations. So I'm going to end there and hope that we have just at least a few minutes for some discussion. Thank you again for having me. We really appreciate this.
Yeah, no, thanks, Grace. And I'm glad you made the point that you did. You know, being from Maryland, I don't imagine that the ocean is much different in Maryland than it is off of South Jersey. And you know, I think your last point, the last point that you made, which was, you know, maybe expanding this to other states, just, you know, it's, I realize your focus is on impacts to New Jersey at this point, but this is a really important issue for the entire Mid-Atlantic throughout the range, as well as New England. So I think additional discussion at some point down the road would be useful to try to expand the work you're doing into some of these other states, so that you know, we can have a better network for the entire region. So that wasn't much of a question, but I appreciate the comment you made at the end. I've got Peter Hughes with his hand up. Go ahead, Peter.
All right, we must be back online now.
Mike, you got me? Can you hear me?
I can hear you. Go ahead, Peter.
Okay. Grace, good morning. This is Peter Hughes. We've worked together in the past on some various projects in regarding to ocean acidification. You had an earlier slide that showed some red dots, and it was kind of a coastal slide, varying degrees of color red, indicating higher pH, going down to a color of orange with lower pH. And it looked to me around the Delaware Bay, there was a pretty high area of pH, and you'd actually identified that Cape May County was kind of, you know, has a risk assessment on the second level, which is pretty high. And I'm wondering if there is, and I know your work, that's the one, and I know your work was New Jersey centric, but I was wondering, are you seeing any similarities with the outflow of the Delaware Bay in comparison to the Chesapeake Bay, in comparison to Long Island Sound, big bodies of water that provide a lot of introduction of fresh water into the ocean environment? And I was wondering if there's any coalition between those, or is your work just, I mean, this presentation is focused on New Jersey, but I was just a curiosity of mine. Thank you.
Hi, Peter. No, it's nice to hear from you. Yeah, there's been a lot of independent studies looking at acidification in those different water bodies. But as far as I know, there hasn't been like a synthesis analysis that has looked at like a comparative analysis between the bays. And I know that there hasn't been one even in the Raritan Bay, which I'm still surprised by. So maybe I just need to get to work and do that. But yeah, I mean, the estuaries themselves have seasonal signals. You know, Chesapeake Bay, I think, is probably the most studied estuary in terms of ocean acidification, and there's been a lot of papers out about that. Wei-Jun Tsai in Delaware, he's a carbonate chemist, has published a lot of papers about acidification in the Chesapeake Bay. And Marjorie Friedrichs at VIMS now has a forecast model that actually includes ocean acidification. It's like a 2-day forecast. And so that's pretty cool that they're starting to kind of have that forecasting power for acidification in the Chesapeake Bay. So I would say the Chesapeake Bay is pretty farthest along in terms of independent studies. But yeah, I haven't seen a comparative analysis, but all estuaries are going to deliver fresh water and nutrients to the coastal system that are going to act in the same way in terms of the carbonate chemistry, or at least a similar way.
Thank you, Grace.
Thanks, Peter.
Yeah, thanks so much, Grace, for that, and thanks, Peter, for your question. Is there anybody else from the council that has any questions for Grace? Is there anybody from the public or from the audience, whether you're participating either in the room or virtually, that has anything they'd like to add at this time? Okay, seeing no hands at this time. So maybe you could go back. Grace, did you have a next steps slide?
No, it's right now we're just trying to, I'm trying to get out of the screen here. Oh, there we go. I'll put it back to where I was.
Yeah, just kind of let's figure out what the next thing, where the
Yeah.
council can, continue to engage, and what, if there's anything that you need from us at this point, or do you plan to update us down the road?
Yeah, so I'll continue. So it's my group is working with NOAA to develop some of the ocean acidification products that are going into the annual State of the Ecosystem report. So you'll be seeing those every year now in the
Okay,
reports.
excellent. That's great.
So that's one way to keep you informed. But specific to this effort, I'm sure I'll probably be reaching out to people independently. We're trying to contact people in the aquaculture industry and commercial fishing industry, finfish and shellfish, to kind of figure out ways we can, add sensors or add measurements to their operations that would help fill in gaps in kind of that we've identified in the observation network. So that's kind of where we're at right now, is just who do we reach out to, and who would be willing to, have a conversation about it.
Okay. Yeah, and working for a state agency, we have folks that do that. I mean, so yeah, please, feel free to, send me anything via email that I could send off to our, it's not something that I do in my role with Maryland DNR, but we have folks that are involved in this, and I can put you in touch with them if you're not already. So that would be great.
Yeah, thank you. Yeah, and I will say for your first comment that you mentioned before Peter asked his question, that I'm on the steering committee for MACAN, the Mid-Atlantic Coastal Acidification Network, and I'm on their science working group. And we are, constantly in touch with all of the other Mid-Atlantic states on their activities, and we're always having discussions on, how can we all each state improve what we're kind of working on. So we know what's going on in each state, and that's been a really nice kind of regional effort as well.
Yeah.
So
Yeah,
and
of course.
we're staying in communication on those things. So
Yeah, it's a really important topic, and we didn't get into it today, but I've heard some reports. I think there's even a few articles written that the acidification, the increase in acidification, is having effects on the development of larval fish. And it's not just the shellfish, but it's just a concerning issue that we need to stay on top of. So I want to thank you for your report today. I do have Dan Farnham with his hand. Go ahead, Dan.
Yep, thank you, Mr. Chair. Can you hear me?
Yep.
Yep. Grace, just a quick question to that last slide you had. As far as the sensors, could they be deployed from a commercial fishing boat, like a longline boat, either bottom or surface longline boat?
yeah, I mean, it would depend on the equipment. I know that there's been, we've been able to put sensors in things like lobster traps, and on like dredges. You can put like a profiling sensor on a dredge. I'm not sure about long lining, how we would do that, but we can always collect just water samples and analyze those for discrete samples, for discrete measurements of things like pH and total alkalinity and different parameters. So yeah, I think it would depend on what equipment is used and how the operations are actually taking place. But those are the types of discussions that we want to have with the industry.
Yeah, I'd be willing to have a conversation with you offline at one point if you want. You know, after we get the email from the council. Either way, I'll be willing to look into it. Thank you.
Great. Thank you so much.
Yeah, thanks, Dan. Thanks, Grace. I really appreciate you being here with us today to present this information, and we'll look forward to continued updates over time. So in the interest of time, I am going to move on. At this time, excuse me, I'm sorry. At this time, I'm going to flip the baton over to Wes Townsend, who is our council's vice chair. And Wes is going to take us through the next two agenda items before we break for lunch. So Wes, thank you very much for offering to stand in for a few minutes. And I'll be here. I'm just going to step back a little bit and let you take the lead. So I appreciate it. Thanks, Wes.
All right, thank you, Mike. Yeah, go relax a little bit. We may have a busy afternoon ahead of us. Let's start with
What's going on this afternoon?
Lunch break.
I'm just joking. Go ahead.
Yeah.
I'm just joking. Yeah. I'm going to get my thoughts together for later.
All right, let's go start with the 2023 Atlantic Surf Clam and Ocean Quahog specifications. Jessica, whenever you're ready.
Thank you, Mr. Chair. Good morning, everyone. I'm Jessica Coakley with council staff, and I'm originally from New Jersey, so I can talk really fast. So I'm hoping we're going to make up some time. So today we're going to talk about the 2023 specifications. Thought we'd make up time if my clicker. There we go. Okay, so for today's review of the 2023 measures, I'm first going to review the surf clam fishery information document. We have, it's basically a fishery data update. Dan Hennon at the Northeast Fisheries Science Center was able to provide us an update there. We do not have any new survey data to look at this year. The survey was not conducted in 2020. And in 2021, there were some challenges completing the survey. So we hope to have the survey completed in August of 2022, and then hopefully for the next review, we'll have some survey data to look at. You last received a level 3 surf clam stock assessment and a level 1 ocean quahog stock assessment in 2020. And at that time, you used that information to set measures for 2021 to 2026 for both of the managed species. So you have measures on the books till 2026. Once I go through the fishery information document for surf clams, I'll review the performance report for both species, go through the staff recommendations, the SSC recommendations, and we'll repeat the same for ocean quahog. So based on that management track stock assessment, surf clam is not overfished because the biomass is above the target level. And overfishing is not occurring because the fishing mortality rates are below the threshold. This is a diagram of the surf clam landings through 2021. It's broken down by area with Southern Virginia, Delmarva, New Jersey, Long Island, Southern New England, Georges Bank, and then any other areas that weren't captured in those categories. So as you can see in 2021, the landings were similar to those in 2020, just a little bit higher, but still a good deal lower than what we had seen in previous years for surf clams. And most of the landings at this time are coming out of New Jersey, Long Island, Southern New England, and Georges Bank, with a small uptick of landings occurring in that Southern Virginia area. These are distribution maps that were provided as part of our data update. The upper left panel is the 1980s, so that shows you that the distribution of the fishery was much further south at that time. And then the top, bottom right panel gives us the data for the most recent few years, including 2021. And you can see most of the fishery at this time is prosecuted off of that New Jersey, Long Island area, Southern New England, Georges Bank. This is the surf clam landings per unit effort, also broken out by individual area. And then the black solid line is the total landings per unit effort for that year across all of those areas. So over time, surf clam has seen a decline in the landings per unit effort, and it's a pretty steady trend, as you can see. In 2021, there were 41 vessels that participated in the fishery. That's 2 fewer than participated in 2020. There were 8 processors or dealers that reported purchases of surf clams and ocean quahog in 5 different states. There was a slight increase in the overall ex-vessel value. It was $24 million in 2021, up $1 million from 2020. And that was largely driven by the slight uptick in landings that occurred in 2021, not a huge swing in the prices. This figure gives the surf clam landings through, with data through May 26. The quota is 3.4 million bushels across the top. The green line is the quota rationing trajectory. So if the quota was taken through the fishing year at a perfectly steady rate, the landings lines would appear on top of that green line. The previous year, 2020, is shown in yellow. And as you can see, the current year landings are about on par with what we saw in the prior year. So the fisheries performance report was developed for surf clams and ocean quahogs this year. It's a combined report. Our advisors met on April 19th, and they were asked the typical series of trigger questions we asked for our performance reports. There's a lot of detail in that report. I'm not going to cover all of it here today. I'm just going to touch on some of the critical issues that our advisory panel identified in that report. I will also note that in the report, advisors indicated they'd like to see status quo quotas because quota stability translates into fishery market stability for them. So one of the topics that came up was related to regulations for shellfish safety and the Food and Drug Administration. So apparently in the last few years, regulations have been updated related to PSP and other public health closures. Advisors have indicated that NOAA Fisheries has not addressed these changes, which would affect their fishing access and their ability to access areas on Georges Bank. NOAA Fisheries, they feel that these safety measures are unnecessary and that NOAA Fisheries and GARFO really hasn't been taking these up in a timely manner. So the advisory panel requests that the council consider sending a letter to NOAA Fisheries and appropriate public health safety groups in NOAA and the FDA to ask them to prioritize this issue. Our advisors also had some highlights related to COVID-19. They noted that sales to restaurants, food service was low for 2020 and in 2021. However, they've really seen a ramp up in food service demand, so they feel like that has been less of an issue in 2022 and will be less of an issue going forward. However, they noted that demand is high, but they've really had limitations in terms of the amount of product that's available. So their inventories, they really don't have a lot of inventory in stock. What they've been getting in has been going out quite quickly, and they've been able to sell more than they've actually been able to produce. So that was an issue that they raised. In addition, they again highlighted the importance of that the council support research in the Great South Channel Habitat Management Area. That's that Nantucket Shoal, Southern New England area. They feel that the lack of access in that area has been a very big challenge for the fishing industry. They'd like to see the council's work on this issue, and they are frustrated with their lack of ability to work through the exempted fishing permit program in order to gain access to that area. So they felt that there were some time components in that access area that should be revisited. So there are some seasonal restrictions for cod. They had concerns about those. They recognized that the council had taken initial steps with that leadership level discussion that took place earlier in the year, but they again requested that the council make this a priority issue and that the council follow up with New England to conduct a cross-council workshop on this issue. In terms of other important issues, the Surf Clam and Ocean Quahog Advisory Panel would like to request that 2 AP members have seats on any fishery management action teams for issues related to this fishery. Oh, and that was the end of the critical issues. So just a quick reminder, for Atlantic surf clam, when we set catch limits, we start with the overfishing limit. The SSC reviews information, identifies scientific uncertainty. You set an annual catch limit for surf clam, consider management uncertainty, and set an annual catch target. So there's a single annual catch limit that's set in this fishery. So for 2021 through 2026, when this process was followed, the SSC identified the overfishing limits and ABCs in the first two columns, and the council identified the annual catch limit, annual catch target, and the commercial quota for this fishery in those last three columns. The SSC reviewed the information that was provided to you in the fishery data update and some additional background, and provided their recommendations for 2023. So at this point, if Dr. Rago is online, Dr. Rago, would you like to speak to this slide?
Okay, thank you, Jessica. Yeah, I'll make it very quick. There was, as you said, there was no new information. The critical part or important part is that the LPUEs in the surf clam fishery have shown these steady declines, but we recognize, of course, that many factors enter into that those changes over time, including shifting areas as well as economic profitability. But we do know that they are consistent with the model-based estimates of biomass, which are approaching BMSY levels. We note, of course, that the catches tend to be below the existing quotas and that stock is not overfished and overfishing is not occurring. So to sum up, the SSC recommended continuation of the previously approved quota for 2023. Thank you very much, Jessica.
Thank you, Dr. Rago. So the staff also reviewed this information, and like the SSC, they recommended no changes to measures for 2023. I'll just note that in order to maintain status quo measures in this fishery, the council would need to make a motion to recommend the regional administrator suspend the minimum surf clam size. It's an annual requirement, but no other action would be required to maintain the same measures. At this point, Mr. Chair, do you want me to continue with ocean quahog, or do you want to pause for questions on surf clams?
Why don't we go ahead and continue, and we'll get questions at the end.
Great. Okay, so ocean quahog, based on that last management track assessment, the ocean quahog stock is not overfished because it's above the target level, and overfishing is not occurring because it's below the threshold level. This is a similar figure to what I showed for surf clams that the center had provided through data through 2021. So for ocean quahogs, you'll see most of the landings are occurring in New Jersey, Long Island, and southern New England in this fishery. The landings in 2021 are just a little bit higher than 2020, but they're still a good bit lower than what we had seen in previous years. This is the ocean quahog landings per unit effort. It shows a slightly different pattern overall than we saw for surf clams. The solid black line, the overall landings per unit effort, has remained relatively flat over time. And this is the distribution map by 10-minute square that was provided as part of that data update. The top left panel is the early 1980s. The bottom right panel is the most recent few years, including 2021. So you can see a shift from the fishery being prosecuted in more southern areas to more northern areas over time. So in 2021, there were 16 non-main vessels that participated in the ocean quahog fishery. 3 main vessels participated in that main mahogany quahog fishery that has a separate bushel allocation that's allotted to it. That's the lowest number of the main vessels we've seen in recent years, so that has dropped a bit. There were 8 processors, dealers that reported purchases of surf clam and/or ocean quahog in 5 states. There was a slight increase in the overall ex-vessel value in 2021. It was 18 million, so it was up 2 million from 2020, and most of that pattern is being driven by that slight uptick in landings. So for the non-main fishery, with data through May 26, the current year landings are about on par with the previous year, 2020 landings at this point during the year. And this is for the main mahogany quahog fishery. 100,000 bushels are allocated to that fishery. The 2020 landings were substantially lower than the 100,000 bushels allotted. And as you can see this year, the 2022 landings are a good bit lower than the prior year. So ocean quahog, the flow chart is slightly different. We go through the same process to set an annual catch limit, a single one for this fishery, but there are 2 annual catch targets that are set in the ocean quahog fishery, one for the non-main fishery and then one for that main mahogany quahog fishery. So based on the information you had in 2020, the SSC set the OFL and ABCs that are shown in those first few columns, and the council recommended the ACLs, ACTs, and commercial quotas that are shown in these last few columns. That ACT, I know, that's the combined ACT for both of those fisheries. So with that, it looks like the webinar is having a little trouble catching up to my slide advancement. There it goes. All right, Dr. Rago, would you like to cover this slide?
Sure. Thank you, Jessica. I may try to make this even shorter. The stock appears to be, continues to be in good shape. It's at about 2 times the BMSY level. And that is supported also by the overall LPUE trend. The model, or the observed fishing patterns, as you've noted, are moving northward and eastward over time. But in aggregate, there are no new signals. There is no information that suggests adjustment to the quota is appropriate. So the SSC recommended continuation of the previously approved quota of 44,082 metric tons for 2023. Thank you very much, Jessica.
Thank you, Dr. Rago. The staff also looked at this information, and like the SSC, they recommended that there be no changes to measures for 2023 in this fishery. And so to maintain status quo measures in this fishery, no action would be required for ocean quahog. And that concludes my presentation. Thank you, Mr. Chair.
Thank you, Jessica. You got through it very fast. Any questions for Jessica or Dr. Rago? All right, seeing none, we do have a motion that needs to be passed so we can maintain the 2023 measures. Can we get that up on the board, please? And while she's doing that, Peter Hughes, you have a question? You want to make the motion?
So the motion, it doesn't appear it copied into the file. But I know the chairman, you've got a draft of it. It's pretty straightforward.
Yes. The motion would be moved to request that the regional administrator suspend the Atlantic surf clam minimum size for 2023.
Jessica, where's that? Where is the motion? I mean, why doesn't it appear on the screen? Yeah, let's get it up there on the screen so that we can have Peter make the motion. And also, while we're waiting for that, we don't have to do anything relative to the specs for surf clams and quags unless we want to change them, correct? So we don't need a motion today.
No, you don't need a motion for any of those other measures. Thank you, Mary.
And do we have a second on the motion? Kate Wilkie seconds it. Discussion on the motion, Peter?
Does the motion need to say request the regional administrator suspend the?
We can add that, yes.
Sorry, we're just running on the fly, so.
Yes, that looks correct now. Any discussion on the motion? Any member of the public want to discuss the motion? Peter, your hand is still up for, oh, you're off now, I said, okay. Is there any objections to the motion? And one abstention from Garfo. The motion passes with one abstention. Thank you, Jessa. Is there anything else that needs to be brought to us? All righty, let's move on to the next topic then. Sharon Benjamin, is she going to be presenting here? Yes, okay. We have a presentation by Sharon Benjamin on the equity and environmental justice strategy. And Sharon, whenever you're ready, we're ready for you. Push the red button.
Now it's red. Okay. And oh, there it is. And it works. Great. Okay, give me one moment. All right. So just some backup notes here. So good morning. Thank you for having me. My name is Sharon Benjamin. I'm part of the NEPA team at GARFO, the National Environmental Policy Act analysis team at GARFO. So I get to work with excellent council staff regularly. I've been at NOAA Fisheries for a few years. I started as a contractor with the Social Sciences branch down in Woods Hole at the Northeast Fisheries Science Center. And then I had the opportunity to work with the Habitat Division at GARFO. And then during the pandemic, I transitioned into my current role with the NEPA team. So I'm here to introduce the NOAA Fisheries Draft Equity Environmental Justice Strategy. So as you know, obviously, our work at NOAA Fisheries affects communities who depend on marine resources and coastal and marine ecosystems for environmental, economic, social, cultural benefits. And the NIMT strategy was written to guide each region in identifying existing barriers to achieving equity for these communities. So I have a few goals for today. I want to provide some context about the Equity Environmental Justice Strategy, or EEJ strategy. I might try to say EEJ so I don't continue to trip over EEJ. I want to go over some of the terms used in the document and share some ways that you can provide feedback on the strategy. So I would note that this presentation is going to be a bit familiar for some of you who participated or listened in to the Council Coordination Committee meeting in May. It's a similar overview to what was presented by NOAA Fisheries Assistant Administrator Janet Coit and Deputy Assistant Administrator for Regulatory Programs Sam Rauch at that meeting. And there was some parallel work going on that Council staff Jose Montañez co-chaired the CCC working group that wrote an excellent report on council work across the country on EEJ issues. So I appreciate the chance to introduce it to folks who haven't heard it yet and members of the public listening in. So for some context, there we go. In January 2021, the Executive Order 13985, Advancing Racial Equity and Support for Underserved Communities, I usually shorten it to just Advancing Racial Equity. The executive order was signed, and in response, the NOAA Fisheries formed a working group that's composed of about 35 members of staff from each regional office and each science center, as well as some program staff representation. And so I'm actually the GARFO representative to that national working group. So our renewed interest in EEJ arises from this executive order and builds on mandates of earlier ones. These executive orders have been enacted to guide NOAA Fisheries and other federal agencies for implementing EEJ into their missions. And in addition, as you know, NOAA Fisheries work is mandated by several other laws, including the Magnuson-Stevens Act, Endangered Species Act, Marine Mammal Protection Act, and others listed here. And all of these laws have components that incorporate the goal of achieving equitable outcomes. For example, with Magnuson-Stevens Act, National Standards 1, 4, and 8. So, forgot about the slide part. So, as I mentioned, I want to go over some terms. So the first one is equity. Equity is defined in Executive Order 13985, and it's the consistent and systematic fair treatment for everyone, including those who belong to underserved communities. And I'll get to the term underserved communities in a second. But this graphic I chose is a simple illustration of the concept of achieving equity through ensuring that different users have the appropriate gear they need to enjoy the benefits of a resource, and in this metaphor of a bike path. And so in the first line, under equality, all of the users have the same sort of bike, whether it suits them or not. And in the bottom line, for achieving equity in outcomes, everyone is able to enjoy a bike path with the appropriate gear they need. So it's a simple metaphor, but I like it because it's got users having an active role, and it's a little different from other illustrations I've seen on this topic. So the next term is environmental justice, and it's a two-part definition. It refers to the fair treatment and meaningful involvement of all people. It's a definition that comes from the Environmental Protection Agency. And simply put, we want to ensure both equal access to benefits and the protection from environmental harm and hazards for all communities. And the last term, there we go. The last term is underserved communities that I mentioned before, and it's also defined in the executive order. There's a lot of text on this slide, but basically it describes groups that have been systemically denied opportunities to participate. There are many groups in this definition, but the bottom line captures it all. For all persons that are affected by persistent poverty or inequality, they're included in this definition. So, as I mentioned earlier, the draft strategy broadly identifies ways that communities may experience barriers to fair treatment and meaningful involvement in our work at NOAA Fisheries. We're thinking with the strategy about how we reach our stakeholders and how our work serves communities that rely on coastal and marine resources. So this list is some of the ways that these groups may not be served equally in our work. So I'll go over them very briefly. The first is a lack of awareness of underserved communities. We need to ensure we know who we're talking about, who needs to access our resources, and better serve them. Structural barriers refers to policies and organizational priorities that may inadvertently reinforce reinforce inequity. Barriers to accessing services include language barriers, time, or money. System complexity. If you don't know how it works, it's hard to be involved. Gaps in expertise refers to the fact that we may need staff with additional specific skills in terms of outreach, language, or cultural literacy to reach some communities. And finally, gaps in representation can create additional barriers to knowledge, recognition, and communication with these communities. So, some examples, this is a simple slide with a couple of examples, but NOAA Fisheries and other agencies have been incorporating EEJ into our work for decades. One great example that was, I believe, referenced earlier was the Social Vulnerabilities Indicators Toolbox. It's been a program developed for a while, but we recognize we need to do more. Our current efforts don't meet the scope and duration of some of the challenges that certain communities are facing. And we haven't had a really formal coordinated strategy to develop an EEJ plan to implement this. So two very simple examples from our region I chose. I chose these because they're simple and easy to explain, but also they're a great way to point out the inward-facing and outward-facing approach we're taking. We're looking at in both directions. So as an example of outward-facing, our programs, the recreational fisheries outreach activities, several materials were translated in terms of recreational boating guides and safe fishing guides, translated into other languages, including Spanish. And an example of an inward-facing effort is to increase diversity in our hiring practices, was building relationships with minority-serving institutions. So the NIMS strategy, as I mentioned, takes a broad national approach and provides a framework for incorporating EEJ work into our daily programming. But it's going to require a step-down implementation plan at each region and each programmatic level. And so we're looking for feedback to improve the strategy. So if you think back at the barriers I just went through a moment ago, these questions on this slide are examples of ways to clarify the barriers in our region related to research and monitoring work is one specific example. And if you read the strategy, and I hope you do, you'll see a lot more detail going into these questions and other questions. But I don't need to read the whole slide for you, but the first one is, who are underserved communities? And it's a simple example, is just how can we better communicate with folks who need our services and make sure we are in touch with the right people. So a little context on how the strategy was developed. The working group was formed in spring 2021, shortly after the executive order. Last fall, we solicited early input from federally and non-federally recognized tribes, territories, and indigenous communities. And we drafted the strategy over the course of a year, that working group of 35 folks, and began to roll it out just early in May this year. So we plan to finalize the strategy later this year and follow through with regional and programmatic implementation plans next spring, excuse me. So we need your help. And we're hoping your input will help us shape the strategy, frame out ways to incorporate EEJ into our daily activities, expand our work. We're seeking comments on both the national strategy, the draft document, as well as any regional specific comments you may wish to offer. The strategy document itself was included in the briefing materials, and if you also want to, you can click through on this slide. The presentation was also provided. And this slide has a link also to the Google form where we're accepting comments. And again, I don't know if I said this, but we're hoping for comments before August 19th, so the end of the summer. There will also be some public webinars later this summer. There was one at the end of May, and there will be some in-person meeting dates that will be announced soon. They haven't quite figured out the logistics there yet. So thank you so much for your time and your attention. To wrap up, NOAA Fisheries has a renewed focus on meaningful engagement with underserved communities, and we look forward to hearing you and your feedback, your thoughts on the strategy, again by August 19th. And thank you for your attention, and I'm happy to take any questions you might have.
Thank you for the presentation, Sharon. Any council members have any questions? Dewey?
Yeah, thank you. And could you go back to your slide? I think it's 7. Your definition of the folks you're trying to reach.
Serve communities. Yeah. It's just a little.
How about one?
There we go.
Okay.
Is that it?
Yeah.
Okay.
I was curious in reading the population sharing in particular characteristics. Seemed like there's a mission of man in there. You have women and girls. I was wondering if it's a man or boys. Wouldn't that be included in there?
Well, the underserved communities refers to groups that have been systemically denied full opportunity to participate. And I believe this list is referring to groups that have historically had barriers to participating in different ways. And I would leave it there. I'm not sure how to expand on the definition. Men and boys are included in other places in the groups listed that are in other minority groups that might be experiencing adversity. I'm not sure how else to expand on the definition, but I'd be happy to follow up and see if I can provide more information at a later time.
Yeah, I was just curious because I was reading over this and just saw, it looked like to me maybe a omission and maybe it's somewhere in the document. Thank you, though.
Any more questions? Anybody from the public have a question for Sharon? Seeing none, thank you. Oops, we do have one.
Thank you.
Just have a quick comment and you have to remember that these EOs are part of a big picture, not just for fisheries related issues, but a lot of things that are going on across the country. So, but I do understand your question, Dewey. And if you go to page 3 of the policy, there is a statement, for example, that indicates that specifically in the fisheries context, this is just an example. Underserved group within fishing communities may include, for example, subsistent fishing participants or the dependent fishing vessels, crews, and fish processing and distribution workers. So I think that from the fisheries perspective, that's kind of a good way to kind of think about this issue.
Thanks, Jose. It's a great example.
All right, seeing no more questions, Sharon, thank you very much.
Thank you.
And we're slightly ahead of schedule, so let's take a little bit longer lunch and we will start the meeting again at 1 o'clock this afternoon. Thank you.